Hello.
Sir, it is
Yes.
audible to me.
Okay, okay. Is there any technical issue still there at modern group of institutions?
Yes,
Is
sir.
it settled there? So I start now? Shall I start now?
Yes, sir.
Okay, okay. So here is Dr. Alok Nahata from Yingzi Agricultural and Industries, Sandiri and Berhad, that is private limited, from Malaysia, and I will be speaking on quality control and standardization of herbal drugs. So coming to quality control for efficacy and safety of herbal products is of paramount importance. Quality can be defined as the status of a drug that is determined by identity, purity, content, and other chemical, physical, and biological properties, or by the manufacturing processes. Quality control is a term that refers to the processes involved in maintaining the quality and validity of a manufactured product. In general, all medicines, whether they are of synthetic or of plant origin, should fulfill the basic requirements of being efficacious and safe, and this can be achieved by suitable clinical trials. So, coming to herbal drugs, the herbal drugs denote plants or plant parts that have been converted into phytopharmaceuticals by means of simple processes involving harvesting, drying, and storage. A practical addition to this definition is also to include other crude products derived from plants which no longer show any organic structure, such as essential oils, fatty acids, fatty oils, resins, and gums. Now, coming to derived or isolated compounds in the processed state, such as extracts or even the isolated purified compounds like strychnine from Nux vomica or mixtures of compounds like heparin from Heparis precatorius are not included in the definition of herbal drugs. So herbal drugs, the medicinally used plant parts of herbal drugs and how much of the plant part contributes to the use as medicine. We can see here that most of the part is contributed by roots, followed by whole plant and bark. Flowers have around 10% of the part which is used as a herbal drug. In general, quality control is based on three important pharmacopoeial definitions. Identity, it's very important. Is the herb the one it should be? And purity. Have to check whether there are contaminants, example in the form of other herbs which should not be there. And then most important is the content or assay. That is the content of the active constituents within the defined limits or not. So it is obvious that the content is the most difficult one to assess, since in most herbal drugs the active constituents are unknown. Sometimes marker can be used which are chemically defined constituents that are of interest for control purposes, independent of whether they have any therapeutic activity or not. Such type of markers we call as phytochemical markers. Now to prove identity and purity, criteria such as the type of preparation, physical constants, adulteration, contaminants, moisture, ash content, solvent residues have to be checked. The correct identity of the crude herbal material or the botanical quality is of prime importance in establishing the quality control of herbal drugs. Now, identity can be determined by macroscopic and microscopic examinations. Now coming to macroscopic examinations, usually we talk about the voucher specimens like when we refer to the herbariums. So voucher specimens are reliable reference sources, but there are some sort of limitations. Like outbreak of diseases among plants may result in changes to the physical appearance of the plant and lead to the incorrect identification. Sometimes an incorrect botanical quality with respect to the labeling can be a problem. I give you an example. In 1990s, a South American product labeled as Paraguay tea was associated with the outbreak of an anticholinergic poisoning in New York. Then when subsequent chemical analyses were done, it revealed the presence of a class of constituents that was different from the metabolites normally found in the plant from which the Paraguay tea is made. So when we refer to voucher specimens, there are certain limitations. Now purity. Purity is closely linked with the safe use of drugs and deals with the factors such as ash values, contaminants, and heavy metals. However, due to the application of improved analytical methods, modern purity evaluation also includes microbial contaminations, aflatoxins, radioactivity, and pesticide residues. Then finally, analytical methods like photometric analysis, thin layer chromatography, HPLC, HPTLC, GC can be employed in order to establish the constant composition of herbal preparations. Content or assay is the most difficult area of quality control to perform, since in most herbal drugs the active constituents are not known. Now sometimes marker can be used in all other cases where there are no active constituents or markers can be defined for herbal drug. The percentage extractable matter with the solvent can be used as a form of assay. This approach is very commonly seen in pharmacopoeias. Now the choice of extracting solvent depends on the nature of the compound involved and might be deduced from the traditional uses. For example, when a herbal drug is used to make tea, the hot water extractable matter expressed as milligram per gram of air-dried material may serve this purpose. A special form of assay is the determination of essential oils by steam distillation. This is entirely different material. This is actually, you can say, a very specific procedure which is only applicable to the determination of essential oils. And when the active constituents or markers are known, a wide variety of modern chemical analytical methods are available, like UV visible, then TLC, HPLC, gas, mass spectroscopy, and a combination of GC mass, LC mass. They all can be used and are now very commonly being used, all these methods. Now several problems which are not applicable to synthetic drugs which influence the quality of herbal drugs. Herbal drugs are usually a mixture of many constituents. The active participants in most of the cases are unknown. Then selective analytical methods or reference compounds may not be available commercially. Third thing, the plant materials are chemically and naturally variable. Just now I gave you the example of the Paraguay tea. Then the source and quality of the raw material are variable. Then the methods of harvesting, drying, storage, transport, processing all have an effect. And most of you must have read in your syllabus the variability in drug activity. That is the most important thing which affects the quality of raw materials. And that's why the strict guidelines have to be followed for successful production of a quality herbal drug. And among them are proper botanical identification, phytochemical screening, and standardization. Now coming to standardization, sometimes talking about standardization just looks like we have to standardize it according to certain parameters. But standardization involves adjusting the herbal drug preparation to a defined content of a constituent or a group of substances or constituents with known therapeutic activity by adding excipients or mixing herbal drugs or herbal drug preparations. Botanical extracts made directly from crude drugs, crude plant materials show substantial variation in composition, quality, and therapeutic effects. Standardized extracts are high quality extracts containing consistent levels of specified compounds, and they are subjected to rigorous quality controls during all phases of growing, harvesting, and manufacturing processes. Standardization is very difficult in case of herbal drugs, and that much is it compared to the synthetic drugs. It is very easy once you have established procedure. Synthetic drugs can be standardized easily, but in herbal drugs, due to variations at every step, there are a lot of difficulties. So then coming to what are the regulatory definitions, what existing for the standardization of dietary supplements. Now since I am working in the area of dietary supplements, nutraceuticals, and sort of cosmeceuticals also, so there are actually different types of parameters required by different countries. So no regulatory definition exists for standardization of dietary supplements. As a result, the term standardization may mean many different things. Some manufacturers use the term standardization incorrectly to refer to uniform manufacturing practices. Following a recipe is not sufficient for a product to be standardized. Therefore, the presence of the word standardized on a supplement label does not necessarily indicate its quality. When the active principles are known, when the active principles are unknown, marker substances should be established for analytical purposes and standardization. Now coming to marker substances are chemically defined constituents of a herbal drug that are important for the quality of the finished product. Ideally, the chemical markers chosen should also be the compound that are responsible for the botanical effects in the body, and most of the time this happens, and then they are called as biological markers. There are two types of standardization. First is true standardization. A definite phytochemical or a group of constituents is known to have activity. For example, in case of ginkgo, it is having 26% ginkgo flavones and 6% terpenes. It is a classic example of standardization. These products are highly concentrated and no longer represent the whole herb and are now considered as phytopharmaceuticals. In many cases, they are vastly more effective than the whole herb. The other type of standardization is based on manufacturers guaranteeing the presence of a certain percentage of marker compounds. These are not indicators of therapeutic activity or quality of the herb. These example you might have seen most of the cases in all your houses. You might be having some herbal drugs or some products from Indian herbal market. There you will see on the label, you will find that they will be say it is standardized to particular percentage of a particular compound. That they don't say that the whole thing is standardized. They say standardized to this much percentage of these active compound. There is most of the case you can find out in when you see now in India. In I think in every household you will find some sort of herbal things, some herbal products, and then when you look at the label, you will find out that they are standardized compared to a specific chemical constituent. Now coming to the parameters for quality control of herbal drugs. These are very general, but I have tried to give them a different look. Macroscopic examinations, microscopic examinations, then foreign matter, ash, heavy metals, microbial contaminants, especially aflatoxins, then determination of pesticide residues, radioactive contamination, and analytical methods. Coming to macroscopic or organoleptic evaluation. Now, organoleptic evaluation of drug refers to the evaluation of drug by color, odor, shape, size, shape, taste, and special features, including touch, texture, etc. Since the majority of information on the identity, purity, and quality of a material can be drawn from these observations, this I consider as a very important factor in preliminary evaluation. Now, for this purpose, authentic specimen of the material under study and samples of pharmacopoeial quality should be available to serve as a reference. If the reference is wrong, then you cannot come at a conclusion about the unknown plant material. So, these evaluation procedures provide the simplest and the quickest means to establish the identity and purity and thereby ensure the quality of a particular sample. If it is found to be devoid of or different from the specified sensory characters like color, consistency, odor, it is considered as not fulfilling the requirements. Now, since this is very subjective, because every person tasting or having testing the particular product will have his or own different judgments. Like he can have a different smell, and the other person testing will feel that the smell is different. So they say they refer to as characteristic odor or characteristic taste. It means that individual to individual to nullify the individual to individual variation, they have come up with a term called characteristic taste or characteristic odor, and this reference is made to the analyst memory only. So no preliminary treatment is necessary for evaluating the sample in this manner, except for the softening and stretching in case of wrinkled and leaves and flowers, and sometimes you have to beat the bark. Microscopic evaluation. Now, a primary visual evaluation, which seldom needs more than a simple magnifying lens, can be used to ensure that the plant is of a required species and that the right part of the plant is being used. At other times, microscopic analysis is necessary to determine the correct species and the correct part of the species present. For example, pollen morphology used in case of flowers and the presence of certain microscopic structures such as stomata, leaf stomata, can be used to identify the plant part used. Although these may seem obvious, but it is very important, especially when different parts of the same plant are being used for different treatments. Very good example is stinging nettle, which is called Urtica dioica, and it is very commonly found in Uttarakhand. It is a classical example where the aerial parts of the plant are used to treat rheumatism and the roots are being used for treating benign prostatic hyperplasia. Now coming to the another important factor, the determination of foreign matter. Herbal drugs should be made from the stated part of the plant and be devoid of other parts of the same plant. They should be entirely free from free from molds or insects, including the excreta and visible contaminants such as sand, stones, poisonous and harmful foreign matter, and chemical residues. Animal matters such as insects and invisible microbial contaminants, which can produce toxins, are also among the potential contaminants of herbal medicines. Macroscopic examination can easily be used to determine the presence of foreign matter, although microscopy is indispensable in most of the cases. Furthermore, when foreign matter consists of a specific chemical residue, TLC is very important to detect the contaminants. Now coming to another important parameter, that is the determination of ash values. Now to determine the ash content, the plant material is incinerated or burnt and the residual ash is measured as the total anti-acid insoluble ash. Total ash is the measure of the total amount of material left after burning and includes the ash derived from the parts of the plant itself and acid insoluble ash. The latter is the residue obtained after boiling the total ash with dilute hydrochloric acid and burning the remaining insoluble matter. The second procedure measures the amount of silica present, especially in the form of sand and siliceous earth. Now this parameter is very important when you don't get any idea with the first three methods. Then we can find out that the total ash and acid insoluble ash is how much is the presence of that in that particular species, and if we have the correct data, we can easily match. Now, very important thing right now is determination of heavy metals. We don't know where we are getting exposed to heavy metals and what we are eating, which is giving us the metal exposure. So, contamination by toxic metals can either be accidental or intentional. Contamination by heavy metals such as mercury, lead, cadmium, copper, and arsenic in herbal remedies can be attributed to many causes, including environmental pollution, and can pose clinically relevant dangers for the health of user and should therefore be limited. A simple, straightforward determination of heavy metals can be found in many pharmacopoeias and in based on the color reactions with the special reagents like thioacetamide and diethyl thiocarbamide, and the amount present is estimated by comparison with the standard. These are very basic methods which are mentioned in pharmacopoeias. And then when we come to more you can say advanced techniques, then the instrumental analysis have to be employed where the metals are present in trace quantities like atomic absorption spectroscopy, then inductively coupled plasma mass spectroscopy, and neutron activation analysis. Since these are very, actually, you can say very expensive techniques, and most of the times, I don't think students can afford for the purpose of their for the purpose of their thesis or their things. So most of the time, the pharmacopoeial methods are being followed. These things are the instrumental methods are mostly followed in industries and especially when they need a strict quality control and where the heavy metals are the major source of contamination. Now coming to microbial contaminants and aflatoxins, it is a very important parameter because medicinal plants are associated with a broad variety of microbial contaminants, including bacteria, fungi, and viruses. This microbiological background depends on several environmental factors and exerts important impact on the overall quality of herbal products and preparation. Herbal drugs normally carry a number of bacteria and molds, often originated from the soil. Poor methods of harvesting, cleaning, drying, handling, and storage may also cause additional contamination, as may be the case with E. coli or Salmonella species. While a large range of bacteria and fungi are from naturally occurring microflora, including the aerobic spore-forming bacteria, which frequently predominate in the herbal drug. Now, there are specifically laid procedures, like laboratory procedures have been laid down by WHO and Pharmacopoeia have also laid down the specific guidelines for these in general, and complete procedure consists of determining the total aerobic microbial count, total fungal count, and the total Enterobacteriaceae count together with the test for the presence of E. coli, Staphylococcus aureus, Shigella, and Pseudomonas aeruginosa and Salmonella species. European Pharmacopoeia also specifies that E. coli and Salmonella should be absent from herbal preparation. However, it is not always the case that these two pathogenic bacteria can cause clinical problems. For example, a fatal case of listeriosis was caused by the contamination of alfalfa tablets with the gram-positive bacteria Listeria monocytogenes. So, it depends which country finds which thing as important. EU finds that these two things should be measured. The other countries may not find these as important. Materials of vegetable origin tend to show much higher levels of microbial contamination than synthetic products, and the requirement for microbial contamination in the EU also allows higher levels of microbial contamination than in synthetic pharmaceuticals. The allowed contamination levels may also depend on the method of processing of the drug. For example, if the final herbal preparation involves boiling with water, then higher contamination levels are definitely permitted. Now coming to aflatoxins, this is a very important category which is usually neglected most of the time. The presence of fungi should be carefully investigated since some common species produce toxins, especially aflatoxins. Aflatoxins in herbal drugs can be dangerous to health even if they are absorbed in very minute amounts. Aflatoxin producing fungi sometimes build up during storage. Procedures for the determination of aflatoxin in herbal drugs are published by WHO. After a thorough cleanup, TLC is used for confirmation. Certain plant constituents are susceptible to chemical transformation by contaminating microorganisms. Now this is a very important thing, like determination of pesticide residues. Usually in our daily talk also, and usually after the COVID episode, most of the people were talking how to disinfect our green leafy vegetables and the groceries, and then how to get rid of the pesticides. All these things actually, when we talk in our normal conversations with each other, we say that to get rid of the pesticide, you just keep it soaked in water for 15 minutes, or you add a pinch of salt to get rid of the pesticides. But the correct scientific, or you can say the documentary requirements are different. Even though there are no serious reports of toxicity due to the presence of pesticides and fumigants, it is important that herbs and herbal products are free of these chemicals, or at least are controlled for the absence of unsafe levels. Herbal drugs are liable to contain pesticides which accumulate from agricultural practices, like spraying, then treatment of soils during cultivation, administration of fumigant during storage. Actually, pesticides cannot be avoided unless and until you shift to organic foods, because when you are buying something from the market, from the vegetable market, it is 100%, it has been treated with some sort of pesticides or some sort of chemical which will prevent it during the cultivation. So samples of herbal material are extracted by standard procedure, impurities are removed by partition, and individual pesticides are measured by GCMS or combination of both of these. Some simple procedures have been published by WHO, and the European Pharmacopoeia has also laid down general limits for the pesticide residues in medicine. Now coming to WHO and this European Pharmacopoeia, for the sake of following the procedures, we should follow. But the thing is that the things laid down by WHO, in most of the cases, in recent episodes, which not from the herbal, but what going around, the things told by WHO, most of the time 50%, you I don't think you need to believe them, because they don't also have an idea what's going on around. So as far as you need to follow the procedures, better follow the pharmacopoeial guidelines. That's the better one. Even you can follow the Indian pharmacopoeia. I will trust Indian pharmacopoeia more than the other pharmacopoeias. Now, determination of radioactive contamination. Although this is considered as a parameter which should be monitored during the measuring of the herbal drugs and all, but I don't feel that this is actually a very important parameter, or you can say the parameter which you need to be afraid of. There are many sources of ionization radiations, radionuclides occurring in the environment, and a certain degree of exposure is inevitable. Now again, WHO, in close cooperation with several other international organizations, has developed guidelines in the event of a widespread contamination by radionuclide resulting from major nuclear accidents. Taking into account the quantity of herbal medicine normally consumed by individuals, they are unlikely to pose any health risk, first thing. Therefore, at present, no limits are proposed for radioactive contamination, but still WHO has to say something, so it has put up something which you need to follow in case of any nuclear accidents. That's it. Now coming to analytical methods. Now this is an important parameter. I think this should be followed when you go for analysis, quality control, standardization. These are very important things which you should follow. even for the students listening there, or for the industry people, or anybody involved in the herbal drug field, you should follow the analytical methods, because this is what we can actually find out directly. When we do it, we know it, that this particular thing is present in our herbal drug, and what is the composition and how much of it is present. We can know everything, or every constituent of the herbal drug or a herbal mixture or a herbal product, by doing the TLC, HPLC, GC, nowadays quantitative TLC, quantitative HPLC, and there are a lot of advanced techniques which are being used. Now another thing is like I read overpressure layer chromatography, infrared and UV visible spectroscopy, then mass GC, and then liquid chromatography used alone and in combination with GC mass, LCMS, MSMS, and nuclear magnetic resonance are powerful tools, often used for standardization and to control the quality of both the raw material and finished product. The results from these sophisticated techniques provide a chemical fingerprint. I repeat again, a chemical fingerprint as to the nature of the chemicals and impurities present in the plant or the extract. Now, for the sake of presenting this thing, it was very good to say that all these techniques exist and you should use these techniques for standardization. But it depends on the availability of these techniques. So far, what I have seen in the herbal drug, you can say herbal drug realm all over the, especially the Indian scenario, as well as where I have seen in other countries also the things. But so far, herbal drug standardization still is in, I should say, the budding stages, and these techniques are not easily available to many of the people who want to do it. Now, still we are actually limited to most of the time HPLC, and then we go up to HPTLC. The higher techniques like GCMS, LCMS, LCMAS, these things, if you want to avail, I think you have to look towards collaborations with other people or industries, or maybe you have to look for other countries where these things are available. And I know many people who have done these things, but they had to collaborate with people in other countries where they got these facilities and finally got their thesis done, and then got their degrees also. So when we want to do these things for the sake of presenting, it's very good, but on ground I feel only few of these techniques are easily available to the student. And I will from this one, from this presentation, I can just convey my message to the organizers, that when you teach your students, especially the pharmacognosy students or the herbal drug technology students, you should actually teach them these techniques. Plus, if it is possible for you to provide an exposure to the students of these techniques especially, then they will definitely develop some skills and they will know about it. Otherwise, the students will be only limited to TLC, HPLC, and finally HPTLC. I don't think maybe higher techniques are available to any further students in India. I don't think so. Maybe some higher government institutions, they might be having some LC mass or GC somewhere, but I don't think students have an easy access to all these things. So just summing up, what are the factors which affect the safety and efficacy of herbs and herbal products? These are include processing, then residual pesticides, competitions, bioburden, then interactions with food, adulteration, heavy metals, then shelf life of drug, physicochemical status, and then the other compounds which affect the safety and efficacy. So quality control, once started, it should not stop. You should always do some sort of quality control when you are evolving a product, and after its beginning, it should never be static. Thanks a lot. Thanks a lot for a patient hearing, all of you. Thanks a lot. If you have
Thank you,
any
sir.
questions or any queries, you can always contact me on my mail ID. You're always
Thank you,
available.
sir. Now I request to the student get their queries clarified. If you have any question, then ask them. Yes.
Sir, please describe the applications of HPLC technique in the standardization of herbal product.
Okay. Now for HPLC, first thing you will need a marker. You need a marker in case of an HPLC. For example, I give you an example of Hello, is it audible? Hello? Yeah.
Yes, sir.
So I give you an example of a mushroom called Ganoderma lucidum. There is a very, you can say, very potent mushroom which is used for lot of pharmacological uses. So we do HPLC of Ganoderma almost every day. I am involved in a mushroom industry, and here I do the HPLC of for Ganoderma every day. But HPLC will give you peaks of different actually compounds present in that particular mushroom. But unless and until you don't have a standard compound which shows a particular peak, then you cannot match it with the compound. For example, in my analysis, I have a standard compound called ganoduric acid. That ganoduric acid will give me a peak at retention time of 10.5 minutes. If my sample is not having any peak at 10.5 minutes, I straight away will reject it. It is not up to my requirements. It is not correct Ganoderma lucidum. It may be actually another species, not the same species which I want. So HPLC is very important, but for that you need a marker. Yeah, for sort of like just to know how many different types of peaks you are having in a particular extract. Yeah, HPLC is good. So that's for like you suppose you are a student of an M.Pharm, or right now you are doing a PhD. So for you, in case of an HPLC analysis, if you have a marker, then your analysis will be actually, you can say, foolproof and sound, that because your marker is also present in your extract, then you can say that yes, this is a particular, you can say that this marker is a phytochemical marker which I have further confirmed with HPLC. You're not limited to HPLC, you can go for HPTLC also. And there is a hidden, you can say, most of the people don't know, maybe, because HPTLC can also be used for a quantitative analysis. It's just with the UV detector you can use it. If you have HPTLC, then that is also very important, very useful actually. That if you have a sample in TLC there, you have you must be knowing TLC and the RF values, right? So at a particular RF value, if you are getting a spot, and if that spot is also present in the HPTLC, you can directly quantify it with the HPTLC instrument available at your institute. You have to just quantify, and you have to I think you have to activate the UV measure, UV analysis of the instrument, and you can easily find out. The instrument itself will give you how much of that particular compound is present in that particular mixture. So these two compounds, these two actually methods like HPLC and HPTLC, I consider them as very important as far as the herbal drugs are concerned. And don't ever underestimate TLC. That is the basic and the most important one. If you know how to do TLC, if you are actually, I should say, a master of TLC, then definitely HPTLC you will be actually, I would say, you will enjoy doing HPTLC and then HPLC afterwards, because the basics are clear. Then you will actually, I will say, you will enjoy the herbal drug scenario if you have all these things. Most of the times nowadays what I am seeing in India is, most of the times they consider the herbal drug criteria most of the time pharmacognosy. I am seeing from here is a bit discouraging for me also. I am a man of herbal drug technology, and that is a very wide scenario, a very wide area where you can do a lot of research. As a matter of fact, most of the research in other branches of pharmacy is also being done taking the herbal molecules now. So I hope I have made your question clear.
Yes, sir. Thank you, sir.
Okay, thanks.
Thank you, sir. It was indeed an invigorating and extremely informative session. Now I request Honorable Professor Dr. Akhilesh K. Singhai, sir, to give his concludatory remark on this session.
आलोक जी नमस्कार.
Namaskar, sir.
Dr. Alok Naheta has covered the topic quality control and standardization of herbal drugs. Actually, very frankly, I will say that this is a topic which can be discussed at least for a week, for continuously for few of us. But he has
Yes, sir.
nicely and very smartly summarize the topic within 40-45 minutes. So, sir, I congratulate and appreciate your efforts to compile the presentation in such a nice way that everybody can understand how the quality control is important for the standardization of drugs. You very smartly defined the quality herbal drugs and how the medicinally the different parts are used for the extraction of the drugs. You have also focused about identity, purity, and assay of the herbal drugs, analytical methods which are used for identification or for assay. You have listed all the methods. You also focus about that how the method of harvesting, drying, and storage can affect the constituents of the drug. There are numerous examples which affect these variables on the quality of the drug. You have also enlightened about that no regulatory definitions are there for standardization of dietary supplements. Types of standardization, you said about that how they can be classified. The different parameters of quality control of herbal drugs, you also defined that. Very well, it has been explained that how the organoleptic evaluation of drugs are there. So you have classified in then different 8 categories, that macroscopic, microscopic, organoleptic, S values, heavy metals, and microbial contamination and radioactive contaminants, how they are important in the organoleptic evaluation of the drugs. You also stated about the different parts of the plant, how they can affect the quality and the component or composition of the drugs in that. Lastly, you concluded about the analytical methods, the different analytical methods like TLC, HPLC, GC, QTLC, combination of GC with MS or MS with MS, and NMR and HPTLC, how they are applicable and how they can be used for the standardization and quality control parameters of the herbal drugs. You also targeted about the chemical fingerprinting of the herbal molecules. And lastly, the conclusion was that there are factors which may affect the safety and efficacy of the drugs. So you very smartly summarized the topic, that how the quality control of the herbal drugs can be done. Thank you, sir. Thank you.
Thank you, sir. Thank you. It was nice. It was really a pleasure, and blessings from you are very important for me.
Thank you, sir, for giving your valuable time for us. I request to Ms. Kratika Sharma, ma'am, to escort Professor Dr. Akhilesh K. Singhai, sir, back to auditorium. Thank you, sir. Thank you, sir. मैम सबको बोलिए ऑन कर लीजिए, कैमरा ऑन कर लीजिए, कैमरा ऑन कर लीजिए। जल्दी ऑन, एक एक करके, एक एक करके। Without consuming any time, I gratefully welcome our resource person, Dr. Nagen Singh Chauhan, a senior scientific officer and government analyst at Drug Testing Laboratory Ayush. May I audible, sir?
Yes, I'm hearing. One second. Is the screen visible?
Yes, sir.
Okay, thank you.
Okay, sir.
Good afternoon, everyone. My topic of presentation is phytopharmaceutical challenges, future prospect in therapeutic activity. नाना औषधि भूतम जगति किचिद द्रव्य मूपल भवते. This is the shlok that are given in the Ayurveda. The meaning is that in this world, not a such drug is available which is not to be used as medicine. In other words, it is said that all drugs may be used as medicine. Another shlok in Ayurveda, Yoga dapi visham tiksharam uttamam bheshidam bhavat bheshidam chapi duryaktam tiksharam samapade visham. Means with proper process and logical use, you can make poison or toxin as a medicine. But if the formulation of medicine is not properly processed, it can act as a poison or a toxin. You can see this is a chart that show how the plant can be used as a therapeutic activity. First, plant used in traditional system of medicine, maybe it's an Indian or a Chinese system of medicine or any regional medicine. Then as previous speaker already said, with the help of extraction and other technique, the isolation of the active constituent and the screening of the plant extract can be done. This is the scenario from 18th century to the current scenario. The various phytochemical molecules that are discovered in 1800, the morphine molecule is discovered in 1816, capsaicin, then quinine. In 19th century, taxol, artemisinin. The artemisinin is the molecule that is discovered for the anti-malaria in the China that I will discuss in later slides. And this is the chart how the journey of the molecule can going on. In world, slide is in the world about 45,000 plant species in the world, in which 15,000 medicinal plant and in which 120 medicinal plants are largely quantity used in the industry. About 800 plants used by the pharmaceutical and cosmeceutical industry, which are most coming from the cultivated source. More than 10,000 licensed pharmaceutical units manufacturing are available, in which 55,000 are the pharmacy, and in Ayush about 5 lakh registered doctors are available. In Indian market, about 1600 herbal formulation are available, and according to WHO, 70% of Indian population extensively use traditional and alternative medicine for their health care. This is the basic chart of the Indian system of medicine, Ayurveda, Unani, Siddharthi, and in which about 90% are from plant origin, and 5% are from animal origin, and 5% from mineral origin. The slide is visible. Excuse me.
Yes, sir.
The slide is visible.
Sir, you can do a slideshow by pressing
I
F5.
already done.
Sir, you can do slideshow by pressing F5
Yeah,
key.
I already running slide. That's why I'm asking.
Sir, it is not visible on full screen.
Okay, one thing. I think now it is visible.
So the screen is visible, but वो full screen visible नहीं हो रहा है. So you can go with the slideshow button on menu bar.
नहीं, now it is visible.
Yes, sir.
Okay. I think those right. Your screen is paused. I think from your side something is happening. Hello. हाँ जी. नहीं आपकी तरफ से पॉज कर रखा है मैडम. नहीं यहाँ पर मुझे यहाँ ऑप्शन दिख रहा है पॉज. हाँ हाँ देखिए. एक सेकंड अगेन है. रिज्यूम शेयर. नाउ लुकिंग. Again I'm sharing. One second again I'm sharing. Share.
Hello, sir. Sir?
Yes.
Sir, you save this presentation in PPSX mode and start again. That will be, you can easily show the slideshow.
One second.
Just save as, you go and save as and then select the PPSX, that PowerPoint slideshow mode. Then it will be easily visible. Won't be that problem.
One second. I will. One second. Please wait. I'm trying to share the slide. Is the screen visible?
Yes, sir.
Okay, 1 second. I will just run it then. Changing. Now visible on second page.
किसके थ्रू?
Hello.
Yes, sir.
It is visible second page.
No, sir, you can do it by arrow key. Just press drop down arrow key.
1 second.
Yes, sir, now it is
Okay,
visible.
okay. I understand. Something technical problem is there.
Yes, sir, yes, sir.
Okay, okay. This slide is previously seen by you? Nana Aushadhi? No? Okay, okay. Sorry for technical interference. Again, I will present my presentation.
Okay, sir.
Nana Aushadhi Bhutam Jagati Kichari Drav Mul Bhate. In this world, not a such a drug is available which is not to be used as a medicine. Means, in other words, it is said that all the drugs may be used as medicine. Another shlok in the Ayurveda that is given above, the meaning is that with proper process and logical use, poison or toxin can be considered as medicine, and with improper use of medicine can be considered as poison or a toxin. Look on the look. This is a chart where you can find the main phytoconstituent, how it can be go to the therapeutic. The plant generally used by the traditional system of medicine, maybe from an Indian system of medicine or Chinese system of medicine, or a local tribes people use plant as a medicine. Then the pharmacognostic and the natural product scientist done isolation of the active constituent or the screening of the plant extract, and further the allopathic scientist or the pharmaceutical chemist scientist, they do the active moiety of the plant and they do chemical modification to make more therapeutic active. These are the few research from the last 20 decades, the various research done by the various scientists. In starting, the name is morphine, discovered in 1803. Further, 1848 papaverine, then pilocarpine, then artemisinin, and this research is going on one by one. In world, about 45,000 plant species are present in India, in which 15,000 are medicinally important, and out of which 120 plants are used in large quantity for the medicinal purpose. In industry, like pharmaceutical, cosmeceutical, about 800 plants are generally used by the industrial person. And in India, about 10,000 licensed pharmaceutical units are available that are based on plant-based medicine. And about 5 lakh Ayurvedic doctor or traditional healers are available registered with Ayush. And according to WHO, 70% of Indian population extensively use traditional and alternative medicine for their health care. And as you know, the Indian system of medicine divided into 3 parts: Ayurveda, Unani, Siddha. The 4th category is also added, that is Sowa Rigpa of the Himalaya region, and out of which in about 90% of the medicine are from plant origin, 5% from animal and 5% from mineral origin. and 1-2% from the animal origin also. This chart show where the remaining use of the plants are available. It is used in the medicinal product, as a nutraceutical, as well as cosmeceutical, as a herbal extract, as a herbal tea. In India, about 8 lakh Ayurvedic drug industries are available, and in Chhattisgarh 32, and in Madhya Pradesh around 850 licensed Ayurvedic pharmacy are available in Madhya Pradesh. Since what are the various challenges that are available to the industry when you are using the manufacturing of the Ayush drug or herbal medicines, since raw material 90 to 95% material are come from the to the traders, and they are collected widely from the unliterate people, so the quality is not known. No information about the place and data of collection. No information about the condition of storage after collection and in transit. No information about the maturity of the raw material, and the raw material may be adulterated willingly or unwillingly. So these are the challenges for the herbal industry, for the how we can use for the product. And this is one of the chart. The current in the ancient time and present scenario, why the quality is very important. During the ancient time, the collection can be done based on the Vedas. The drugs are collected by weight in a smaller quantity in proper season, time, and fresh collection. While in current, on the basis of the demand, the drugs are collected by unskilled labor and in huge quantity, as and when required, used for longer period. During the ancient time, they were weight collected in a smaller quantity, and they know the test during the processing process, and because they make a smaller lot for one patient or two patients or about 20 patients. But nowadays, the unskilled labor will be used in the processing, and the large quantity of the drug are made, so no test at all. So there is a large lot of transportation done from all over the world. So this is a major challenge in the quality control of medicine, of the phytopharmaceutical. As the previous speaker already detailed about the quality control, but I have given this chart that adulteration can be done by intentionally or unintentionally. Intentionally means sometimes similar looking material like Ashoka and potash. Both drugs look like similar. So this type of adulteration began. Sometimes exhausted product, the clove, cardamom, the volatile oil is extracted by the industry, and they can sell this drug to the market. Similarly, look like plant like Chirata, Ratanjot, black pepper. They can be adulterated. Sometimes partial adulterate with similar looking substance, the saffron. Sometimes imitation material like clay. And sometimes unintentionally adulteration. For example, common vernacular name. The name of us in India, in Chhattisgarh, some drug is different and some in South is different. Same plant with different vernacular name. So these are the problem in the herbal medicine. True identification is a major problem in current scenario. And what are the challenges and opportunities for the pharmaceutical and health scientists? In current scenario, about 2,000 registered acute and chronic diseases are available, and only in which 15% are curable. And the rest can be treated only symptomatically to greater or lesser effect. And about 3 lakh plant species are present, in which 13% are investigated scientifically. So there are lots of opportunity available for the natural product scientists to do work. And from last 2 decades, there are lots of bioactive natural products are going on researchly. In current scenario, in the COVID scenario, you also look that the acceptance of the herbal product are increases traditionally. From the last 15 to 16 years, about 500 molecules are discovered, in which only 3 get international approval, First molecule is virgin that contain green tea extract of Camellia sinensis and that are registered by Medigen Germany. The second brand is Flujac that is obtained from the plant latex of Croton lacheri, and third one is cannabis that are registered in 2018 and is used for epilepsy. So you can imagine from the last 15 years, only 3 molecules get approval. So it's a very long and tedious process. But as Nahataji said, the botanical and dietary supplement market is increases so much because they only the structure and functional claim, no therapeutic claim. This is the data for the sale of botanical supplement, that is $8.2848 billion. Of the currently used medicine, 40% derived from the natural product and analog, and in the oncology, in the cancer and infectious disease, the natural product molecule is about 70%. The reason of use of the bioresources is that due to the structural diversity, novelty, complexity of the natural molecule, to synthetic to organic chemists to design and modify natural molecule to more potent drug. Now I will discuss the Indian regulatory aspect. In India, for the drugs, these 6 departments are working parallelly for the regulation of the herbal drugs in India. First is Ministry of Health and Family Welfare, in this CDSCO, DCGI, DTAB, and the Ministry of Ayush. Ayush only for the drugs for classical and proprietary medicines. Ministry of Chemical Fertilizer, in which Department of Pharmaceutical, NPPA, Ministry of Commerce, Patent for the herbal medicines and product, Controller General of Patent, Ministry of Science and Technology, in which DBT and various CSIR labs are involved in for the drug regulatory system, and the Ministry of Environment. Environment clearance to the manufacturing side. All these 6 ministries are involved for the drug regulatory system in India. Now, to consider what about the current scenario for the Indian regulatory for the plant drug and healthcare product. So currently it is divided in 2 ministries, Ministry of Health and Ministry of Ayush. The molecule that I will discuss today, that is phytopharmaceutical, it is a part of Ministry of Health, not the Ministry of Ayush. So in the Ministry of Health, 4 parts are there: NC and generic product, that are natural, semi-synthetic and synthetic product. Second one is biological and biotech product. The new third category that is included in 2015 is phytopharmaceutical, bioactive, purified, standardized fraction, and fourth one is dietary supplement that is updated in 2016. And then in Ministry of Ayush, only the Ayurvedic product that are classical or proprietary medicine of ASU drug, Ayurveda, Siddha, Unari. This is the brief history of the Indian system of medicine. In 1995, it is called Indian System of Medicine and come under Ministry of Health and Family Welfare. Later, it is renamed as Ayush in 2003, and from 2050, the independent Ministry of Ayush is created by the Government of India. Before discussing the phytopharmaceutical, I just want to clear that ASU drugs are different from the phytopharmaceutical. This is a new category and it will come under allopathic drug inspector regulation, not in the Ayurveda. So what come under ASU plant drug? So ASU plant drug defined in the Drug and Cosmetic Act in chapter 4A of ASU medicine, and that is divided into 2 parts: classical preparation and proprietary medicine. Classical preparation are those that are defined in the Ayurvedic textbook, 54 textbook under Schedule A. Schedule A, and the proprietary medicine means the rational combination of the ingredient based on the experience of qualified Ayurvedic physicians and each of the ingredient is proposed composition must be in the Schedule 1 of the Drug and Cosmetic Act, and it cover about near 2300 plants. Hello? Can I start?
Yes, sir.
Now the phytopharmaceuticals. The phytopharmaceutical is a new term that are introduced by the Government of India in 2015, and it will come under the allopathic system of Chapter 4 of the Drug and Cosmetic Act, while Chapter 4A deal with the ASU drug. The definition, the drug and cosmetic does not include the word like TM, traditional medicine, herbal drug, herbal medicine, and phytomedicine. In India, only the two systems are running, allopathic system and ASU drug. Why? The chapter phytopharmaceutical will come under allopathic system. While ASU drug include comfort medicine, ethnobotical tribal plant of the drug. The phytopharmaceutical rule come on 30th November 2015 in the gadget number GSR 9182EB that define phytopharmaceutical drug include purified and standardized fraction with defined minimum 4 bioactive or phytochemical compound qualitative or quantitatively of an extract from medicinal plant or its parts for internal or external use for human being. So important parameters that minimum 4 bioactive or phytochemical compound should be there, then it can be defined as phytopharmaceutical. This rule will divided into the two parts. Part 1 that deal with the basic informations and human and clinical pharmacological information, and part 2 that can be generated by the person's manufacturer. First we discuss the part 1. The part 1 include basic information include route of administration, dosage, therapeutic classes. That include the various information like the previously reported traditional use, their side effect, their human or clinical pharmacology information, the various scientific report published, various pharmacodynamic parameter available, the description of the plant in various pharmacopoeia, monograph if available, that can be included in the part 1. And part 2 that is generated by the manufacturer included into 12 parts. First one is the pharmacognostic description. Then that is already the part that quality specification, the various physiochemical parameter like microbial load, aflatoxin, chromatographic parameters, estimation of marker, that all included in the pharmacognostic description. Second one is the quality specification of the phytopharmaceutical at each and every stage, during the raw material stage, during the processing stage, during the packaging stage. Then the formulation of pharmaceutical and drug applied for, then the various excipient detail, their quality specification, all that be included in the formulation area. Now the fourth one is manufacturing process of formulation. That is the outline method of manufacture, dosage form, in process, quality control, packaging step, and final packs. Fifth one is stability data. Sixth one is the safety and pharmacological information available. 7.1 is the animal and toxicity data and safety data. It includes rodent and non-rodent species as per Schedule Y, in vitro genotoxicity, dermal toxicity, teratogenesis and 8th one is the clinical trial as per Schedule Y, phase 1, phase 2, phase 3. All this, then 9th one is regulatory, 10th one is the marketing information, and 11th one is post-marketing surveillance. So these are the 11 points in the part 2 that is covered under the phytopharmaceutical, and this data can be generated by the manufacturer before applying to the license of any new phytopharmaceutical. In the next slide, I will give the example the various phytopharmaceutical available. Now for the lipid, hyperlipidemic, the garlic have various phytoconstituents that have anti-hypercholesteremic activity, and the red yeast rice that contain colistin 3 have shown also the very anti-hypercholesteremic activity. So nowadays the scenario is changing. People are going toward the taking medicine that are obtained from the natural product. Various categories, the various plant that are used for purgative, senna, trifla, carminative, carum carvi, gingiva, cinnamon, antidiarrheal, Holleria antidepressiva, punica granatum, hepatoprotective, edographlite, eclipta, curcuma, Tinospora cordifolia. In current scenario, last COVID-19, the Sun Pharma with collaboration with ICGB and CSIR, they have discovered going to work on the phytopharmaceutical drug that are obtained from the aqueous extract of Cocculus hirstus, and they just published their case report, their clinical study data, and they have found that this aqueous extract of Cocculus hirstus have good therapeutic activity against COVID-19 molecules. So what are the challenges to use the use of that phytopharmaceutical? We have to develop a mechanism to recognize and not to block the traditional use. At the same time, assuring the quality, safety, and efficacy of the medicinal plant. And there is need to engage in workshop and training course and exchange of information related to the phytopharmaceuticals. These are the few books that I have published related to the phytoconstituent, novel drug delivery of phytoconstituent, and the natural product in vector-borne disease management. I end my seminar with this word: Om Sarve Bhavantu Sukhina, Sarve Santu Niramaya, Sarve Badani Pashyantu, Ma Kashyudu Kamavite. Thank you.
Thank you, sir, for enlightening us with your valuable, motivational, and amazing session. Am I audible, sir?
Yes.
Yes, sir. Thank you so much, sir, for your valuable words.
Any question?
If you have any question, raise your hand. Yes.
Good afternoon, sir.
Good afternoon.
When a phytopharmaceutical drug is considered as a new drug,
Yes, it is a new drug and it come under allopathic system,
yes, sir.
not in Ayurveda,
Okay,
ASU
sir.
system.
Thank you, sir.
Anyone else?
Thank you.
Sir, what are the phytochemical challenges occurs during the treatment of coronavirus?
Coronavirus. As you know, as you are very alert, during the COVID-19, we all are at home, and we have found that the Ministry of Ayush, they have generated some guideline to take kada or take some Ayurvedic preparation. So in India, as I see, those persons that take regular the Ayurvedic medicine or the some take the observation of the Ayurvedic guideline, they have very good health. So I think not much challenge, but in current what happened, the regulatory have some problem. When you this is a disease that come first time, so regulation problem is there to accept the various data, because we need lots of study, lots of clinical data needed to accept the phytopharmaceutical. But if in the future, if any such type of disease come, I just show you the slide in that this slide. Aqueous extract of Cocculus histus. They have found good result in the COVID-19. So work is going on, because the disease not go from anywhere from us from last 5 to 10 years. So we have to prepare for our immunity, and this type of formulation have very good help. As you are living in the MP, I say you, this plant is easily available in everywhere in MP, Cocculus histus. And they also collect plant from Madhya Pradesh for the research purpose also.
Thank you, sir.
Thank you.
Thank you so much, sir. Now, I'm extremely happy to welcome our Honorable Eminent Speaker, Dr. Hanumantachar Joshi, Professor and Principal, Sharda Vilas College of Pharmacy, Mysuru. Please take a session, sir.
Well, good afternoon to everyone. And मुझे पता है आप पूरे भूखे हैं. It's almost 1:45. My talk was scheduled to be for 40 minutes. I'll cut it down to 20 minutes. And I assure you that I will not make you boring. Okay. I have traveled all the way from Mysuru to talk to you and give some information regarding it. Can I have the first slide? See, my topic is, the presentation of today's topic is, what is the use of herbal pharmacotherapy in healthy aging? आप सब जानते होंगे एजिंग का मतलब क्या है. अभी छोटे हैं आप, फिर भी one day they will become aged, isn't it? अभी छोटे हैं, बाद में आप एज बन जाएंगे. So what do you mean by healthy aging, and what is the role of herbal pharmacotherapy we will be discussing today, with a special reference about a disease which is called as Alzheimer's disease, right? आपने शायद सुना होगा Alzheimer's के बारे में, right? एक बार आदमी जब भूल जाता है तो पूरा वो भूल जाता है. Can I have the first slide? Is it possible? Yeah. So that's what. See, I will be having two parts in this. One is what is healthy aging, and why do we call it as a healthy aging? देखो, जब आप जवान होते हैं तो ठीक सब ठीक ठाक होता है. जैसे ही आप 25 के बाद 30 के बाद होते जाते हैं तो क्या हो जाएगा? Slowly will get develop problems regarding your memory, isn't it? So for example, before he starts the slide, let me tell you. कभी-कभी क्या होता होगा? देखो, मेरा हिंदी इतना अच्छा नहीं है, लेकिन I hope you will manage it, right? See, sometimes what will happen, you want to go out of your room, isn't it? You want to come out to your college, and suddenly you feel that you are not carrying, the key, the bike key or the car key, whatever it is. Okay, you will start searching everywhere. आप सारे घर में ढूंढना शुरू करेंगे. बाद में आपको पता चलेगा आपके पैकेट में आपका की है. होता है कभी-कभी, बहुत बार होती है. इसको हम बोलते हैं amnesia. What is amnesia? It is a reversible loss of memory. Okay. So मैं क्या बात कर रहा हूँ? I am talking about cognitive disorders, which is very, very important, because you have to start planning from at the age of 25 to 30. Once you become aged after 60 or 70, it will be very, very difficult, right? ये दूसरा slide है. Okay. Yeah. So there are two things. One is amnesia. Other one is dementia. तो मैं वही बात कर रहा था amnesia के बारे में. आप भूल जाते हैं, बाद में आपको याद रहेगा. That is amnesia, which is very, very common. और दूसरी बात क्या है, कभी-कभी you want to go to your refrigerator at home, you go there and आपको suddenly क्या हो जाता है, आपको पता भी नहीं चलता आप क्यों आए हैं यहाँ पे, है कि नहीं? And then you will realize that you want to take some ice creams or you want to take some some vegetables, something like that. That is amnesia, which is totally reversible in nature. Reverse हो जाता है. And what is dementia? डिमेंशिया में क्या हो जाता है, एक बार आप if you forget something, the entire thing is completely forgotten. One example, let us try to imagine, देखो आपका brain है, isn't it? Brain में we have got neurons, isn't it? Neurons are the functional units of brain, isn't it? तो लगता है कि किसी ने फॉर एग्जाम्पल ब्रेन में कुछ लिखते हैं, फॉर लिखने का मतलब है याद रखना, तो कोई एक पेंसिल लेके वहां पर लिखता है, लेट अस इमेजिन लाइक दैट, है ना? आप अपने ब्रेन में कुछ लिख रहे हैं पेंसिल से, तो आपको याद है वो, लेकिन सडनली क्या हो जाता है, कोई आपके एक इरेजर लेके, रबर लेके पूरा उसको इरेज करना शुरू कर दे तो क्या हो जाएगा? सिमिलरली इट विल हैपन इन डिमेंशिया, इट इज टोटली इरिवर्सिबल इन नेचर. एक बार आप भूल गए तो सारे भूल जाएंगे, आपका नाम भी भूल जाएंगे, आपका फादर का, मदर का, सब कुछ. एंड देखो वन थिंग इज वेरी वेरी इंपॉर्टेंट, जो भी हम हैं इट इज बिकॉज ऑफ मेमोरी. आप जरा ध्यान दीजिए, आप जस्ट इमेजिन कीजिए, यदि आप भूल जाएंगे आपका नाम, आपका परिवार का नाम, खाना खाना या दवाई लेना, तो क्या हो जाएगा? Absolutely there is no life. So that's why we call it as we are our memories. Can I help you in this? I think I should help you. लेकिन ये तो खुल रहा है, आप सारे बंद कर दो, ये नहीं है, मैंने इंदौर का दिया था वो ओपन करो. आप पूरा बंद कर दो, बंद करो सारा, पूरा बंद करो. अभी आपने ओपन किया था, डेस्कटॉप में सेव किया क्या आपने? विंडो में जाओ, ओपन करो वो. नहीं, एक मिनट दे दो, मैं करके देता हूँ. आपको ये शेयर किया क्या आपने? हाँ, शेयर करा. तो फर्स्ट स्लाइड ये करो. फर्स्ट स्लाइड करो. फाइन. सो आई विल बी टॉकिंग अबाउट हर्बल फार्माकोथेरेपी. आपने सुबह से सुना होगा फार्माकोग्निस के बारे में, मेडिसिनल प्लांट्स के बारे में, राइट? सो माय पॉइंट इज नॉट दैट. इवन though I am a cognizant man, my little work was in the industry in the beginning, then I am an academician now. And we work on purely on the the formulations, right? Sections लेना, उसका आइडेंटिफाई करना हम करते थे पहले, लेकिन पूरा रिसर्च जो है it is for Alzheimer's. So I'll be talking about two parts here. One is neurodegenerative disorders and what is the role of herbal pharmacotherapy. आप सब जानते हैं, next slide. आप सब जानते हैं what is herbal pharmacotherapy, isn't it? Therapy is nothing but treating something by using medicines. What is herbal pharmacotherapy? Using the herbals. And you can ask me a question. जब अल्जाइमर्स की आप बात कर रहे हैं तो we are talking about herbals. Why not the synthetic drugs? The problem is we don't have any medicine till now, right? अल्जाइमर्स के लिए मैंने आपको अभी डिटेल बताया, we don't have any medicine till now in the world for complete cure of it. We have few drugs in the market like donepezil, rivastigmine. I'll be telling you in the next slide, right? But they have their own side effects. So first one is what is healthy aging? 2 minutes I'll just spend on that. See, number one is include survival to a specific age. What is aging? एक एज तक एक आदमी को रहना है, and as per the data what we have in India, what is the average lifestyle of a human in India? How much? 60? You are near. What are the exact now? 65. One more guess? Yeah, it's almost between 65 to 69. The slide also I'll show you, right? And being free from chronic diseases. Healthy aging का मतलब है सारे chronic diseases like diabetes, then hypertension, other things, the person should be free from that. And well-being, good quality of life, right? से मैंने आपको जो बताया, याद नहीं रहना है. The person has to be dependent upon other people. It is not called as a good quality, isn't it? जो भी अभी बच्चे हैं आप पढ़ क्यों रहे हैं? To get a good job, isn't it? And why you are working? To get money, to have a good life. तो ये सब करने के बावजूद, if your quality is not good, life quality is not good, nothing can matter, right? And high social participation and only mild and cognitive impairment. जैसे एज बढ़ता जाता है तो क्या होगा? आपका मेमोरी थोड़ा घटता जाएगा. लेकिन आपके मतलब if you are very young and if you have a problem, we have to be very very careful about it. Okay, so this memory impairment is also a problem. That's why I have taken the topic that is neurodegenerative disorders. 3 terms are there. One is generation. What is the meaning of generation? कुछ grow होना, isn't it? For example, बाल क्या है? वो generate होते हैं. जब काटेंगे क्या होगा? Regenerate होगा. isn't it? One example we will take, for example, skin we have, skin cell. हम कहीं जा रहे थे, थोड़ा सा क्या हो जाएगा, मतलब थोड़ा सा injury हो जाएगा, and you will get the skin once again. But the main that is called as regeneration. What is degeneration? Completely मतलब जाना. And what is neurodegenerative disorders? These are the disorders which are connected with the neuron. आपको ये याद रखना है, देखो आपका hair भी एक cell है और आपका skin भी cell है. ये regenerate हो सकते हैं, but neurons can never regenerate. That's the problem there, right? Next one. Next. Yeah, कैसे मतलब how to go proceed with healthy aging? Normal thing. We should have a nutritional food. तो आपको याद में I am talking about aging. You are younger people, but you will become aged sometime, isn't it? And I am talking about the physical aging, but not the mental aging, isn't it? Yeah. See here, nutritions are very much required. आपको मन में लगता होगा कि सब लोग खाते हैं nutrition, but our data says even very educated people who can afford, they don't take nutritional food like other countries. They lack in vitamins and all, right? तो इसमें ध्यान देना बहुत जरूरी है. Next one. Hello. Hi. Next one. आप जरा यहाँ पर concentrate करो, बात तो होती रहेगी बाद में. Yeah. So this is brain. थोड़ा सा जाना जरूरी है, right? And you know very well, right? What is the size of the brain, a human brain? Of a cauliflower. आपके गोभी मंचूरी बनती है ना, जैसे गोभी का, right? Your structure looks like that. Okay. And how many neurons are there? You can see there are 100 billion neurons and 100 trillion, the synapses. And I don't have a pointer, but you can see in hippocampus is very important in the brain, right? And what will happen to the brain? Next one. What will happen? अभी क्या करेंगे? We will imagine a orange fruit, right? संतरे, isn't it? We have a fresh orange and we have kept in the open air. तो क्या होगा इसका? कुछ देर के बाद क्या होगा? Shrink होता जाएगा, isn't it? The tissues will start shrinking. Same thing will happen in the brain also. As age progresses, आप कुछ करो या कुछ ना करो, क्या करना है मैं end में बताऊंगा आपको, right? Shrink होता जाएगा, and what will happen? It leads to degenerative disorders. One example is Alzheimer's and dementia. Okay. And how do we identify what are the symptoms and what are the warning signs for it? सबको आपको लगता होगा, है ना? सबको लग रहा होगा कि मुझे भी हो सकता है क्या? Because मैं हर दिन भूल जाता हूँ. पढ़ना भूलना amnesia नहीं है. समझ गए ना? Like आपको एक structure है ना? Like vitamin B12. यदि आप मुझे पूछेंगे सर आप इतना बात कर रहे हो vitamin B12 का structure देख के दिखाओ, मैं नहीं जानता, मैं भूल चुका हूँ. When I was a student, like I used to remember and I just wrote it, I passed the examination and I forgot it. लेकिन आप मेरा area of research के बारे में बताएंगे तो कुछ हम बात कर सकते हैं नहीं? It's very common. Yeah, what is the number one? See, what are these disorders? These are the mental disorders characterized by various, the impaired cognitive functioning and involved in disturbance in memory, as I told you, and brain is either damaged or injured, right? इसका मतलब ये नहीं कि हमेशा brain shrink होना चाहिए. कभी-कभी एक shock से, for example, ischemia, I think you know very well. What is ischemia? Ischemia is, the less concentration of oxygen in the brain, isn't it? जब ऑक्सीजन का मात्रा कम हो जाती है ब्रेन में क्या हो जाएगा? स्ट्रोक होगा, isn't it? So that also leads to all these disorders. And dementia and amnesia. Just now I told you, what is amnesia? Can anyone? Amnesia is a reversible loss of memory. एक बार भूल जाते हैं तो बाद में वो समझ में आ जाते हैं. What is dementia? It is irreversible nature. एक बार भूल जाएंगे तो कभी याद नहीं आएगा. Next one. Go quicker. Yeah. So this is a very next. This is a very progressive disorder. Progressive का मतलब है ये बढ़ता जाएगा, right? It will keep on continuing till the end and click करो, right? It is mainly because of the disease of the brain. It changes the behavior, mood and personality. आपको कभी-कभी लगता होगा कि कोई आपके teacher, excuse me, I am also a teacher, so don't think in another way, as a friendly manner. कोई आपको टीचर बोलता होगा, देखो यार मुझे मूड के ना पूरा खराब है. हम भी सोचते हैं, देखो आज मेरा टीचर का मूड बहुत खराब है तो हमें बहुत होशियार रहना है. ये कोई बड़ी बात नहीं है. The problem is there in the brain of the person. हमेशा मूड स्विंग्स होना, है ना? जल्दी गुस्सा हो जाना, हमेशा गुस्सा रहना, लोगों को डांटते रहना. There is a problem in the brain. And what's the problem? I think you would have studied about neurotransmitters in the brain, isn't it? Brain में कुछ neurotransmitters होते हैं, like 5-hydroxytryptamine, epinephrine, right? Dopamine, acetylcholine. ये जो न्यूरोट्रांसमीटर जो है वो हमारा पूरी बॉडी मेंटेन करता है। जैसे कि आप हैप्पी है ना, यदि आप एक मॉल चले गए, आपने कुछ नया देखा तो आपको बहुत मजा आता है, या नहीं तो आपको कोई ओल्ड फ्रेंड आते हैं, आप मिलते हैं, ये आप नहीं, आपके न्यूरोट्रांसमीटर है। सो देयर इज अ चेंज इन दैट लीड्स टू ऑल दिस कॉम्प्लिकेशन। एंड अदर वन इज द accumulation of the proteins in the brain। बॉडी में क्या होता है, ब्रेन में there is a protein called as beta amyloid protein in the brain। क्या है इसका नाम? beta amyloid protein in the brain। it leads to all this complication। next one, next, next। so we कभी कभी क्या होता है, people बोलते हैं ना, लोग बोलते हैं कि ये memory problem is a very common thing, but it is not a common thing। it's a very very serious one। I will tell you in the next one। right now, why I'm talking about Alzheimer? Because most of there are so many types of dementia। what is dementia? I'm telling once again, it is an irreversible loss of memory। and why I'm talking about Alzheimer's disease, it's mainly because it is in higher the prevalence इससे ज़्यादा होता है। next। okay, this is what happens in the brain। I told you, we took an example of a orange and we understood that what will happen। see my left side, you can see the healthy brain which is very intact। and in case of say very AD, it starts shrinking, hippocampus undergoes degeneration, right? and all the parts of the brain undergoes degeneration, which is called as neurodegeneration। next। I think we'll skip it। next। yeah, what are the risk factors? Number one is smoking, right? and other one is diabetes mellitus, hypertension, which are very very common। जब एज होता जाता है तो अभी क्या हो जाएगा, there is a prediction by 2050, right? almost 80% of Indians will be diabetic, right? There is already a lot of diabetes is already there because of the food, because of lifestyle and everything। it's one of the major risk factor। next। and why it is called Alzheimer? एक साइंटिस्ट था 1910 में, उन्होंने इस डिसीज का एक नाम दिया था, right? उसका नाम है अल्जाइमर, इसलिए अल्जाइमर्स रखा गया है। next। yeah, and what are the warning signs? number one is memory loss, right? चलो, आपको हफ्ते में एक बार आप भूल जाते हैं कि ठीक है, हर दिन भूलते हैं तो दिक्कत है, समझ गए ना? और लोगों का नाम भूल जाना, चेहरे भूल जाना आते आते फ्यूचर में, है ना? and these are some of the warning signs। and next one is the decreased judgment। कभी कभी क्या होता है ना, जब लोग जाते हैं कोई मॉल में कुछ खरीदने के लिए, I will wind up in 15 minutes, don't worry, right? yeah। and क्या हो जाता है ना, they will be asking other person, तो मुझे ये ड्रेस लेना है क्या, मुझे ये करूँ क्या? दोस्ती में चलेगा, लेकिन for everything if you are having a problem in judgment, it's a brain disease, right? and another one is previous और पीछे जाइए, बहुत आगे आ चुके आप। राइट। देन प्रॉब्लम्स विथ लैंग्वेज। जब हम बात करते हैं, जो दो वर्ड्स के बीच में राइट ठीक से क्या है ना वर्ड आना चाहिए। फॉर एग्जाम्पल मुझे प्रॉब्लम हो रहा है हिंदी में, कोई प्रॉब्लम नहीं, बिकॉज हिंदी तो मेरा मदर टंग है नहीं, है ना? एंड एब्सोल्युटली नॉट इंग्लिश, बिकॉज इंग्लिश इज नॉट अवर मदर टंग, इट्स ए फॉरेन लैंग्वेज। यदि आपके मदर टंग में आपको वर्ड्स यूज करने का दिक्कत हो रहा है, तो इसका मतलब है देयर इज अ प्रॉब्लम इन द ब्रेन। हाउ डू यू ओवरकम? कीप स्टडीइंग अबाउट इट, कीप रीडिंग इट एंड बी वेरी वेरी क्लियर व्हेन यू टॉक अबाउट इट। स्ट्रेस में नहीं बोलना चाहिए, राइट? दैट्स व्हाई टू अंडरस्टैंड। एंड अदर वन इज लॉस ऑफ इनिशिएटिव एंड देन प्रॉब्लम्स विद एब्स्ट्रैक्ट थिंकिंग, मिसप्लेसिंग, मैंने बोला था। एंड कभी कभी क्या होता है, मेन प्रॉब्लम क्या है डिमेंशिया में, ये लोग, वन एग्जाम्पल है, वो टॉयलेट चले जाते हैं लेकिन क्लीन हो क्या ना वो भूल जाते हैं, विच इज ए वेरी वेरी सीरियस वन। अभी तो हमें थोड़ा सा फनी लगेगा, लेकिन थिंक ऑफ द पर्सन, राइट? वेरी वेरी पीपल दे वुड हैव लेड देयर लाइफ, इतना मेहनत करके, काम करके, जिंदगी में अच्छा होके ऐसा फेस कर रहे हैं, तो देयर इज अ प्रॉब्लम। नेक्स्ट, या फॉरगेटिंग अपॉइंटमेंट्स, कन्वर्सेशन। ये पेशेंट्स क्या करते हैं, ये बोलते हैं आपने कभी बोला ही नहीं था। मैं पॉलिटिशियंस की बात नहीं कर रहा हूँ, पॉलिटिशियंस कभी याद नहीं रखते ना जो बोलते हैं, और कभी कभी आप दोस्त में भी भूल जाते हैं, हमने बताया नहीं कभी, वो तो ठीक है, लेकिन some important thing we have to remember। and repeating stories, एक बार बोल दिया उसको रिपीट करना हमेशा, again and again। उन्होंने आपको पूछा कि आपका खाना हो गया क्या, आपने बोला हाँ दादी हो गया, वो फिर पूछेंगे आपका खाना हो गया क्या। so these things happens because they keep on forgetting, right? next one, okay, I think he is also very hungry, I can understand that, yeah। and getting lost, कभी कभी क्या होता है, ये लोग बाहर निकल जाते हैं घर से और वापस नहीं आते, because they will be wandering, they will never remember their home place also। next, this is very very clear, driving, banking बहुत मुश्किल है। next one, changes in behavior, blaming the things, है ना? किसी ने दूसरा कुछ कर दिया। कभी कभी पेशेंट्स हमारे पास आते हैं, है ना? every Saturday I will be having some consultancy, apart from my basic pharmacy teaching, we have associated with other organizations, right? हम कर सकते हैं pharmacy वाले. तो क्या होता है, जो सास और बहू होते हैं ना, सास हमेशा कंप्लेंट करती है बहू के ऊपर that she is putting something in my food like poison or something, but they are very good, but that kind of things are coming. और कुछ लोग बोलते हैं मेरे कान में कोई आके कुछ बोल रहा है. So these things happen in the dementia. Next one. Next. Right? And this is the average life of an Indian as far as 2019 concerned. किसी ने 60 बोला, 65 बोला. It is almost 70 years now. That is the average lifespan of a person in India. It is mainly because of our pharmacy people. Because we have made the drug affordable, right? People can easily purchase the drugs now, right? Yeah. Next. Next. Yeah. What are the drugs we have? There are 3 modalities are there. Keep on clicking. Yeah. One is curative. There is no cure for it. Next one is disease modifying. We don't have any drugs for disease modifying. And the third one is symptomatic treatment. Yes, we have drugs. Next. What are they? They are the donepezil, rivastigmine, galantamine. तो क्या होता है, फार्मेसी वाला है जानना जरूरी है. ब्रेन में एक ट्रांसमीटर से न्यूरोट्रांसमीटर है, उसका नाम है अस्टेलकोलिन, राइट? अस्टेलकोलिन एक ठीक मात्रा में सेक्रेट नहीं होती है तो क्या हो जाएगा, मेमोरी का प्रॉब्लम हो जाएगा. तो हम क्या कर रहे हैं, वी आर गिविंग ए ड्रग कॉल्ड एज असेटाइल कोलिनेस्टरेज इनहिबिटर. तो ये क्या करेगा, असेटाइलकोलिन का मात्रा बढ़ाएगा, कहाँ पे, ब्रेन में. देन व्हाट विल हैपन, इट लीड्स टू कोलिनर्जिक टॉक्सिसिटी. तो उसमें क्या होगा, जो पेशेंट होते हैं दे विल स्टार्ट मिक्चुरेटिंग इन द बेड इटसेल्फ. मिक्चुरेशन का मतलब है यूरिन पास करना, टॉयलेट पास करना, वो बेड में करना शुरू कर देते हैं. इट इज ऑलवेज चैलेंजिंग देयर, पीपल डोंट वांट टू डू इट, इजंट इट? या नेक्स्ट, दिस इज द donepezil, which is very wide known. Next. Then rivastigmine. Now it is available in the form of patches, TDDS, transdermal regulatory systems. You can put it behind your neck or on the forearm. It can give you sustained release of the drug. Next, galantamine. But whatever it is, all of them are having very, very serious side effects. देखो, number one उनको याद नहीं है. They are already suffering from a lot of problems. When they start, passing the urine, passing, the stools in the bed, what will happen? It becomes very difficult for them, right? Next. Go for the next one. Yeah, I will skip it. Next. So this is about, the herbal medicines, very old, right? हरप्पा मोहनजोदड़ो के टाइम, हरप्पा सिविलाइजेशन जानते हैं ना, उसी टाइम पे जो मरते थे उनके साथ क्या करते थे, वो कुछ पौधों को भी रखते थे. उनका मन में मान्यता ये था कि जब वो अगली बार आएंगे तो उसको यूज करेंगे. So they are very old. Next. Yeah, there are these things I will touch and I will go for the next one. See, whenever there is a disease, what will happen? Directly we go for drugs, isn't it? It is not actually. We have to go for 4 things. One is ahara. What is ahara? Food. By modifying our food style, right? If it is possible to control a disease, it's very good. It can be possible. One example is diabetes, isn't it? Instead of taking so many drugs, if you modify, your food, it is possible to, cut down the doses. Other one is, which vihara. When you are not able to treat with ahara, then you go for vihara. What is vihara? Picnics. Earlier, सारे हिंदुस्तान में या कहीं भी दुनिया में वो पिलग्रिमेज जाना बहुत जरूरी है ना? पिलग्रिमेज या पिकनिक क्यों जाते हैं? वो छोड़ो, रिलीजियस के बाद छोड़ेंगे, है ना? मैं भी सुबह महाकालेश्वर गया था और यस्टरडे मैं ओंकारेश्वर भी गया था, बहुत अच्छा लगता है. लेकिन सबसे ज्यादा क्या होता है, आपका ब्रेन रिलैक्स हो जाता है, राइट? तो क्या होता है, विहार करने से क्या हो जाएगा, न्यूरोट्रांसमीटर्स की मात्रा बढ़ेगी बॉडी में and it will make you very, very immune. And another one is it will be good for the disease. Other one is vichara, thinking, right? All because of negative thinking, all because of unwanted thinking, isn't it? It leads to a lot of complications, and if we can modify it, it is really good. And last one is aushadha. When you are not able to manage with these 3 things, right, then we have to go for a disease. That's what we learn, isn't it? Next. Next one. I should go for next one. Yeah, so this is one slide. I will not go through the entire slide. Please look at, the number 9. हमें memory loss कब होना चाहिए पता है? At the age of 81 to 90, है ना? तो इसमें आयुर्वेद में भी उन्होंने दिया है what drugs can be taken. Like example is Brahmi and Mandukuparani. So this thing can be. But what's happening at an age of 38 also? जब हम research paper लिखते थे, I'm talking about around 20 years ago. We used to write that by 2025, number will become 5 million in country, India. लेकिन अभी क्या हो जाएगा, number बढ़ चुका है. So that's what I'm trying to say. It is fastly progressing. Next. So these are few of the drugs. I need not tell you. I'll share my slides with your faculty here. You can see whenever it is required. A lot of drugs are available in Ayurveda, right? And Unani, Siddha, All drugs are available. Next one. These are some of the work what we did in our laboratory, like Psittacardifolia, then asparagus. Next. अब जाते रहो मैं बताता हूँ. Next. Yeah, this is one thing I would like to tell you. जो डिमेंशिया होती है, जो डिसीज होते हैं ना पूरे हमारे बॉडी में, 13% डिसीज जो होता है, दे आर ड्रग इंड्यूस्ड. हमने कुछ दवाई लिया होगा, वो क्या करते हैं, डिसीज पैदा करते हैं. वन एग्जांपल इज आवर डिमेंशिया. वन एग्जांपल आई विल टेल यू. यू नो एंटीहिस्टामाइन्स, इजंट इट? जब भी आपको थोड़ा सा क्या होता है, एलर्जिक होता है ना, when you want to, when you are going somewhere outside, right, you will get sneezing. What you will do, you will take what you will take, cetirizine, chlorpheniramine maleate, Monteclair sodium, all these are called as antihistamines. इनको regularly लेने से क्या हो जाएगा, आपको dementia हो जाएगा, right? Even diabetes also, 8% of diabetes is because of the drug induced disease. These are called as drug induced diseases, right? This is one slide I wanted to show you. Next one. Next. And this you know very well, curcuma. जब सारा दुनिया, the entire world was facing difficulty during corona, COVID, who came to the rescue? Curcuma, our own Indian system, right? Next. I'll just go fast with this because all this you know very well. You're already hungry, I know. I don't want to make you, because you're children, I don't want you to be hungry. But I am also helpless. I have been waiting from morning for it, right? Yeah, इतना दूर आके lecture दे जाना तो अच्छा नहीं होगा ना? That is what. So one is ginger, you know very well. Apart from your regular adrak ki chai, right? This is very good for memory. Please keep on eating. Eat fresh ginger. Next. And this is Withania somnifera. आपको जरूरत नहीं है. But when you become more than 45, you can put in your diet, right? Till now I have visited around 37 countries, and I am one of the researchers working on Alzheimer's in the country. Normally when I had been to Japan once, उनके पास एक मेनू होता है होटल में, जैसे हमारा भी मेनू है ना, जैसे कि गोभी मंचूरी, वो बैंगन का भरता है ना, ऐसे ही उनका एक अलग होता है, वो मेडिसिनल फूड है. डायबिटीज लेके अलग है, उनके लिए अलग है. So this is one of the content in that. But otherwise this drug is very well known. It is called as ashwagandha. Next. Next. Allium cepa. What is allium cepa? Right? आप जानते हैं, right? Onion खाइए. कौन सा onion खाना चाहिए? आप खाते होंगे. कौन से? It's a fresh onion. Next. Allium sativum, garlic तो खाते ही हम लोग, है ना? लेकिन ज़्यादा खाना भी दिक्कत है. You should take only one, what I can say, petal of it, not more than that. ज़्यादा लेंगे तो क्या हो जाएगा? Acidity हो जाएगा. It will mimic like your heartburn. Especially aged people के लिए जरा ध्यान से देना चाहिए, नहीं तो क्या हो जाएगा? They will have heartburn. Again, you will take them to the hospital, and they will put ECG and all. They will loot your money, right? You have to be very careful. Next. Aloe vera, right? It is well known. People were using for cosmetics, but it can be taken also as a juice, right? Next. This is our neem, है ना? वो तो पत्ते तो खाते ही है, right? So this is really required. And once in a week, right? You must take, the leaves of Ajada recta. You must chew it, right? And you must spit it out. Okay. Next. Emblica officinalis, it आमला, हम तो खाते ही सब लोग, है ना? So we have to keep on using it. यदि आप अब शुरू करेंगे, तो कम से कम 10 साल आपको डिमेंशिया होने का चांसेस एडवांस हो जाएगा. नेक्स्ट. मेंथ अवेयरनेस, है ना? क्या है? पुदीना, है ना? पुदीना टिक्का नहीं है, लेकिन पुदीना चाय सबको पीना चाहिए. नेक्स्ट. Tulsi, we know very well, isn't it? Tulsi is a very well known drug, but many people are not taking it. But it is recommended that at least 2 times in a week. कितना लेना चाहिए? 3 leaves. 3 होते हैं ना? 3 साथ में लेना चाहिए. Next. Piper nigrum, right? This you are already using. Okay. Next. And Rauwolfia serpentina. Next one. Cassia fistula. It is widely available in the country, but people are not using it. You can take it as a powder. You can just take a powder, make it as a herbal drink. Next. Ricinus communis. यदि आप आपके parents को पूछेंगे या आपके दादाजी को पूछेंगे, वो क्या करते थे? Every once in a week they used to give it for taking it. It is called as the I don't know what you call in Hindi. I have forgotten. Right? This is very good for health. Right? It's a very good cathartic and purgative. Main problem aging में क्या होता है? Constipation. Right? Instead of giving cathartic and purgatives, you can give this. It will be useful. Right? Next. Terminalia arjuna. It is also useful. Next one. Coriander. आप बोलते होंगे कि मैं coriander पत्ते हम हर दिन डालते हैं. वो ठीक है, लेकिन आपको यूज करना है कोरिएंडर सीड्स, fruits, right? They are very much useful. How do you do that? Just take a powder and drink it as you know a herbal tea. Don't tell me that I am putting in masala, right? It is not going to be useful, right? This is going to be useful. Next. Turmeric, you know very well. We have been studying about it. Okay. Next. And fennel, ये तो आप खाते होंगे, right? लेकिन अब थोड़ा सा कम हो गया है खाना, लेकिन अब spit नहीं करना है आपको, पूरा खाना है, right? Yeah. But problem क्या होता है aged people में, they cannot chew and swallow it. तो क्या करना पड़ेगा आपको, उनको एक टी बना के देना पड़ेगा, right? You can just soak it in the overnight. कल, I mean next day in the morning you can give it to them, right? Yeah. Next. Yeah, this is about the herbal drugs which are very being widely increasing, especially after Corona. The production and as well as the market is highly increasing. Next. Same thing. It's about the Ayurvedic drugs. It is highly increasing. Next. And this is the pan. I did not tell you, right? Pan sabko khana chahiye, but not sweet pan, not jarda also, right? There is a content here. आप देख लीजिए, because it is very nicely planned by our ancestral people, isn't it? So this has to be regularly consumed, but not in the classroom. Okay. So when you go home after the food, you can take it. Next. Same thing. So finally, अब तो ड्रग्स के विद ड्रग्स मैंने बोल दिया, विदाउट ड्रग्स क्या करना है, राइट? आई विल जस्ट टेक 2 minutes and I'll wind it up, right? Number 1 is the exercise the brain. Body exercise तो हम कर रहे हैं ना, walking कर रहे हैं, exercise कर रहे हैं. How do you exercise the brain? By playing some brain games, right? Like Sudoku, right? And some brain games are available. You can definitely do that, right? It will be very much useful. And very important is reading. कभी भी आपको minimum दो pages पढ़ना चाहिए. Every day before you go to bed, minimum 2 pages. I am not talking about your curriculum. Other than that, है ना? Curriculum तब पढ़ेंगे. उसके अलावा something new आपको पढ़ना पड़ेगा, क्योंकि brain में जाएगा and your brain will become very active. And next one is keep socially active, not on WhatsApp or not on Facebook, right? Meeting people regularly, physically, है ना? वो बहुत जरूरी है. And other one, go back. I am just ending. Go back. Right? And if the person is smoking, quit smoking, and alcohol in moderation, right? Yeah, I know you would have, because you are pharmacy students, you know very well what is nicotine. It's a CNS stimulant. ये क्या करेगा? CNS को stimulate करेगा. What is alcohol? It will depress the brain. Normally क्या होता है, जो drink करते हैं alcohol, उसके साथ smoking भी करते हैं. तो आप just imagine कर लो क्या होता होगा brain को, right? Something is stimulating, something is depressing. So it is completely challenging the brain. It's very difficult. Don't do that. Okay. And have a healthy diet and have some kind of fish oil. Next. Yeah, this already told you. There is something puzzles, right? Like game, sensory stimulation. Next one. You have to use the brain or you have to lose the brain. That's why we have mentioned it as use it or you lose it. Right? So हमें हमेशा हमारा brain को active रखना है and remain socially active and involved in some kind of voluntary organizations. Next. Yeah, stress से जरा दूर रहना है. Next. And have some kind of hobbies like music or something, right? Curriculum तो हमेशा रहेगा, लेकिन कम से कम 1 से 1.5 घंटा हमारे लिए हमको spend करना चाहिए, right? Next one. Next. अब तो आप बदल नहीं रहे हैं, जब मैंने बोला बदल रहे थे. चलो next. नहीं हो रहा है, तो wonderful. So this is what I wanted to share with you, and I wish you a very wonderful, what I can say, चलो ठीक है, fine. So I hope I have not taken more of your time. I hope it was not very boring, and I am very sorry I had to go very quickly because I also know very well I am also hungry like you, right? And please be in touch with that. And only one call, I know there will be no much questions. If there is 1 or 2, I will take it. But more than that, please keep yourself very active, right? हर दिन minimum 2 पेज पढ़ना है. हो सकता है तो अपने मदर टंग में, है ना? Don't be very stylish, है ना? ये जो अंग्रेजी पढ़ना, वो करना, because we are also very good in English. But the problem is we cannot think in our foreign language. So आप इसे याद रखिए. And I wish you a very good, all the best. And I'm very nice to see, very young people here. Thank you so much.
Thank you, sir. Now I request to the students, if you have any question, raise your hands. There is no questions. Okay. Yes, Prince.
सर मेडिटेशन से न्यूरॉन्स को रीजनरेट किया जा सकता है क्या?
थोड़ा जल्दीबाजी में मैंने उसको टच नहीं किया, अदरवाइज इट वुड बीन वेरी इंटरेस्टिंग एंड वेरी लर्निंग टाइम फॉर यू. प्लीज सिट डाउन. तो meditation is very much important, and like pranayama we have pranayama, because what will happen, it will soothe the brain. soothen होता है. For example, especially corona time क्या आपने practice किया नहीं किया पता नहीं है, but pranayama is very much useful. Lot of work has been done, and even myself I had done that work when I was in Dehradun working earlier. I started my career from Dehradun, North India, and now I am completely in South India now. We had done one work with Himalayan Institute of Medical Sciences with AIMS. तो एक ग्रुप बना के चेक किया जाए तो मेडिटेशन डेफिनेटली हेल्प्स. नंबर 1, it soothes the brain and it will really make your brain active, and definitely it will help here. Actually what we do for our patients is we keep on telling them to do meditation. Not only meditation, even reciting the mantras, है ना. So whatever may be your religion, Hindu, Muslim, Christian, whatever it is, you must be having some kind of mantras that you can recite, right, without seeing. And the songs, but you are very young people. I normally we tell the very aged people, पुराने गानों को देखे बिना याद रखना और गाना. So all this will be very much useful.
Thank you, sir.
Thank you.
Anyone else? No? Thank you, sir. It was indeed an invigorating and extremely informative lecture. Thank you so much, and thank you for giving your time. And now I request to Ms. Kritika Sharma ma'am to escort the Dr. Hanumantacharya Joshi sir back to the auditorium or proceed for the lunch. Thank you, sir. सारे स्टूडेंट्स